Hello, my dear friends. Welcome to Live Prep. Myself, Dr. Gajpati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on YouTube channel and also on our app. But test series can be given only through our app. Now, let us go with a new chapter for the revision, and the chapter name is Excretory Products and their Elimination. And please remember, here also we are confining ourselves to NCERT content only. So, quickly we will revise what is the NCERT content. Clear? And now, here we are talking about what are the various types of excretory products. In general, majority of the excretory products are nitrogenous waste materials. That's why mainly we are talking about three types of nitrogenous waste materials. They are ammonia, urea and uric acid. So these are the major types of excretory waste materials. But not only these, there are some other types of excretory materials are there like water. So why water is coming here? I will tell you. Water, there are some minerals and even some gases like CO2 are also considered under excretory materials. Why we are considering this? Because in different organisms, remember one thing, so some organisms are living in hypotonic conditions, some organisms are living in hypertonic conditions, and depending on their surrounding conditions, sometimes these organisms they are accumulating some water, some organisms are accumulating ions to maintain some concentrations, and sometimes during that ingestion of the food or liquids they are taking more and more quantity of water or ions. In those cases also accumulation of water and ions are also taking place. So in those cases that excess amount should be eliminated, that should be removed. So in some organisms even water and ions are also coming under excretory materials because of this particular reason. This is what about the water and minerals. And now we will talk about the nitrogenous waste materials. How exactly these nitrogenous waste materials are produced? See, every organism for its survival, it is taking some food. And in those food, different types of food materials are present. And upon utilization of these food materials, some byproducts will be released. And these byproducts, now they may be converted into some other products and finally they will be eliminated from the body. Understood or not? So in this way, mainly we are talking about three types of nitrogenous waste materials. This is one thing. And anyhow, what are these three nitrogenous waste materials? And regarding this, slowly we will go with. And under the under the excretory products itself, in the NCERT, even they are talking about CO2 also. Why they are talking about CO2? When we studied about breathing and exchange of gases, so there we studied about CO2. Yes or no? So with respect to human, if you are talking, so you can easily understand, that's why I am talking this example. So we are taking oxygen, and where this oxygen is utilized? In the electron transport system it is used. So this oxygen is used. And already I told you that in the Krebs cycle CO2 is released. So in this way tissues are utilizing oxygen and they are releasing CO2, and this CO2 is released in the large quantities. Now this excess CO2 should be removed. It should be removed. So that's why under the excretory products and their elimination, even we are talking about CO2 also. That's why in the NCERT, in that first sentence, first two sentences, they are talking about some list of excretory materials, those which are harmful to our body and those which are more present than required. Now they should be eliminated. Now all these will be excreted from the body. Now in this list they are talking about ammonia, urea, uric acid, CO2, water and even some minerals also. Yes or no? That is the reason why they have given that list. So you need to understand why they have given these many and why they are coming under excretory materials. That relation also you need to understand. Yes or no? So with respect to water, once again I will tell you, the organisms that are living in hypotonic conditions, now by osmosis what happens? Excess water will enter into the body. Now it will be accumulated. Now compulsorily it should be removed, or else the osmotic balance will be disturbed, right? Same is the opposite case. Opposite is the condition for hypertonic condition. And in those conditions, here what happens? Water loss will happen. So to reduce the water loss inside the body, some materials will be stored. and as part of this some ions also will be stored. So that is what the condition is. So you need to interlink that concept, then only you can understand why they are accumulated. When they are excess than required, then the excess quantity will be released. Yes or no? This is what they are talking here. Now we will focus on this particular three words: ammonia, urea and uric acid, and these are all nitrogenous waste materials, and some organisms are releasing the ammonia as the major nitrogenous waste and they are called as ammonotelic. Some organisms are releasing urea mainly, please remember urea mainly, that's why they are called as ureotelic. Some organisms are releasing uric acid mainly and they are called as uricotelic organisms. And there is a table also in NCERT they have given here. Ammonotelic, under this we are talking some examples of many bony fishes, aquatic amphibians and aquatic insects. Simply NCERT examples are enough, so remember that. Ureotelic, they are excreting urea mainly. And what are the examples? Mammals. Mammals means human is also part of this. So that's why human is ureotelic. Please remember this. And many terrestrial amphibians and even marine fishes. So why they need, why they are mainly talking about the terrestrial organisms? See, in the case of organisms that are living in the water, so even living in the water is already available, and ammonia is highly toxic, and for its removal more and more water is required. So that's why majority of these are living in the water. So you can understand this. But with respect to ureotelic and uricotelic, you can understand that mainly these are terrestrial forms, even though some are related to here. Some are amphibians are there, some marine fishes are there, but still here the condition is something different. And here what they are doing is the water availability is little bit less in general. I am not talking about the marine fishes, in general I am talking. Water availability is less. In that case, the ammonia cannot be the excretory material because it needs more and more water for its removal. So that's why to conserve water, now here they evolved a different type of nitrogenous waste material. Uric acid is the least toxic, so that very small amount of water is enough for this. But urea is moderately toxic, so a moderate quantity of water is required for its removal. So in this way, three important types of nitrogenous waste materials that we are talking. Accordingly, we are classifying the organisms. For ureotelic, examples are mammals, including human, many terrestrial amphibians and marine fishes. Next one, uricotelic. Uric acid is excreted by these organisms. What are these? Reptiles, birds, land snails and even the insects. So under this example of insects, you can also talk about cockroach. So please remember this. This is what the information regarding this particular table. Clear? And now before continuing further, here they have given what are the various organisms, what are the various excretory structures. But before directly going into this, we need to talk one important information regarding these three: ammonia, urea and uric acid. In NCERT, in one paragraph they have given this particular information. So now let us quickly go through this. Ammonia, urea and uric acid. Okay, so just now I told you about the toxicity. Now ammonia is highly toxic, whereas uric acid is least toxic, so that urea is moderately toxic. So since ammonia is highly toxic, for its removal maximum quantity of water is required, means more water is required. And uric acid is least toxic, so for its removal very less water is enough. That's why generally uricotelic organisms excrete this uric acid in the form of a pellet or a paste. But in the case of urea, some moderate quantity of water is required, not as much as required in the case of ammonia removal. Is it clear? And in the case of this ammonia, some organisms are releasing ammonia as it is. But in the case of aquatic, mainly they are aquatic organisms, right? So this aquatic organisms. So in the aquatic means that H plus ion is present in the water. So that's why they are releasing this in the form of ammonium ion. So this ammonia is excreted in the form of ammonium ion. This is another important point you need to remove. Clear? And that too, since it is ammonium ion, it can be easily diffused across the body surfaces. So if you are talking about those bony fishes, so now at the gills itself, this can be easily diffused. That's why for the removal of this ammonia in the form of ammonium ion, kidneys are not playing any role. Directly at the body surface or at the gill surface, now this ammonia is freely undergoing the diffusion. But technically for its removal, large quantity of water is required. Clear? Because it is highly toxic material. This is what one important point that they are discussing. So ammonia means you are writing NH3, ammonium ion means you are writing NH4 plus. Clear? With this information is completed. And anyhow, just now we have studied what are the examples. Now we will talk about the urea. Ammonia means you are writing NH3, but urea means how you will write. Urea is NH2 CO NH2. This is urea. Please observe carefully. NH2 CO NH2. And how exactly this urea is produced? Even in these organisms also, even in the ureotelic organisms also, actually this NH3 group is released. NH3 group is released. And this NH3 group, so two NH3 groups plus CO2, it combines with CO2. Observe carefully. So that now this ammonia in combination with carbon dioxide is ultimately converted into urea. NH2 CO NH2. And then from this two hydrogens, so now they are released as H2O. So in this way, in the case of ureotelic organisms, this ammonia, which is highly toxic, is converted into a moderately toxic material, that is urea. At least toxicity is reduced. So this is what exactly happening. And this particular process generally happens in liver of these ureotelic organisms. So that now within this liver, this urea is produced, and from this liver now it will be released into the blood. Now it will be released into the blood, and from this, through this blood, now it is going to the kidneys. It is going to the kidneys, and now these kidneys, now they will eliminate this particular urea. Now they will eliminate this urea. This is one important point. And there itself they have given one more point, and that point is applicable even for human also. So we are saying that human is ureotelic. Human is ureotelic organism, so that we are excreting urea. But technically, if you are analyzing the composition of human urine, you will find ammonia, urea, uric acid. That means all these three important types of nitrogenous waste materials are released. Not only this, there are some other nitrogenous waste materials like creatinine. Likewise, there is a big list. So there are different types of nitrogenous waste materials. When human is excreting all these three, then why you are saying that human is ureotelic? Because among all these, urea is the major one. Urea is the major one. That's why human is considered as ureotelic. Understood or not? Even though human is excreting all this, still human is considered as ureotelic because the major form of nitrogenous waste is urea. That's why I am saying that even though some other forms of nitrogenous waste are present, based on the major form of nitrogenous waste, we are classifying these organisms. This is the point that you need to understand. Clear? This is what that they are saying. And here they have mentioned one more point. In some organisms, including human also, those which are ureotelic, for this only we are talking, so observe carefully. So those which are ureotelic, in these organisms also, some organisms are retaining some small quantity of urea. That means entire urea is not excreted. Still some amount of urea is stored in the body. In the body means mainly in the case of kidneys. So they have to maintain the medullary interstitial osmolarity. Anyhow, that is part of this particular chapter. At that time, once again I will tell you. So please remember, small amount of urea is retained in the kidneys to maintain some osmolarity, because urea acts as a solute. Urea acts as a solute. When solute is accumulated, the molarity will increase. Yes or no? So in this way, within the medullary interstitium of kidney, to maintain higher concentration, higher osmolarity, small amount of urea is required along with NaCl. In NCERT very clearly they mentioned NaCl and urea are very important to maintain higher concentrations of medullary interstitium of kidney. Yes or no? This is what another point that they mentioned. So this point also you need to remember here itself. So generally many students they know what is ammonia and what is urea, but they don't know what is uric acid. You need to remember this one also. What is the interlinking question? This also you need to understand. Now observe carefully. What is this uric acid? This uric acid has the basic purine ring structure. Already in molecular basis of inheritance, we studied about the basic purine ring structure, yes or no? So here also the basic purine ring is present. And what is the numbering for this one? Numbering is 1, 2, 3, 4, 5. This is 6 and this is 7, 8 and then 9. Yes or no? And what is uric acid? Uric acid is 2, 6, 8. 2, 6, 8 trioxypurine. Uric acid is nothing but 2, 6, 8 trioxypurine. That means at the second position there is a double bond O. At the sixth position there is a double bond O. At the eighth position there is a double bond O. And remaining positions you can satisfy with double bonds. This is what the structure of this uric acid. And please remember, you don't need to by heart this structure, but what is the interlinking point you need to understand. Uric acid is also having basic purine ring structure. That means what type of molecules will form uric acid? It is the adenine and guanine. Please remember this. Adenine is 6-aminopurine. Guanine is 2-amino-6-oxypurine, and these two are the purines. Please remember, adenine and guanine are the purines, and these two are forming the nitrogenous waste that is called as uric acid. Adenine and guanine are the purines that already we studied in molecular basis of inheritance and even in biomolecules also. And these are the purines that are forming the waste material that is called as uric acid. So please remember this. There is a relation here. In this way also they can ask this particular question. So please remember this. Clear? And these are the three important types of nitrogenous waste materials. Uric acid is least toxic. For its removal, very scanty, means very little amount of water is enough. That's why these organisms, uricotelic organisms, are excreting this waste in the form of a pellet or a paste. This is some basic general information that you need to compulsorily remember. Clear? With this basic introduction is completed regarding this different types of nitrogenous waste materials. Now this big list also we have completed. Now they are talking that in the case of higher organisms, now kidneys were developed. But in the case of lower organisms like invertebrates, kidneys are not present, and here they are having different other types of excretory organs. Simply in the NCERT they have given some list, and simply we are reading this. What is the name of that organism, example, and what is the type of excretory structure. At least this also you need to remember. They can ask in the pairing, they can ask in the matching, so huge questions are possible. So remember this. Platyhelminth is like flatworms, and in that they have gave example, specific example like planaria also. In this way they have given this. Any of these names they can ask, so remember this. And even the rotifers and some annelids, please remember, here we are talking about some annelids, but earthworms and other annelids, they are having nephridia, but only some forms of annelids they are having this protonephridia. So please remember this. Sorry. And the cephalochordates, example they gave Amphioxus. So these are some examples they give, and these organisms they are having protonephridia or flame cells they are having. And what is the role of these protonephridia or flame cells? Mainly they are involving in ionic and fluid volume regulation. That means they are involving in osmoregulation. This point was also mentioned in NCERT. So please remember this. And in the case of earthworms and some other annelids, now they are having nephridia. Okay, and here they are involving in removal of nitrogenous waste materials, and also they are involving in maintenance of fluid and ionic balance. Next one, most of the insects, including the cockroaches, they have Malpighian tubules. These are the main excretory structures. Along with this, many other things are there that already we studied in the cockroach lesson. But anyhow, these are the main excretory organs, main excretory structures, Malpighian tubules. They are involving in removal of nitrogenous waste materials, and they are also involving in osmoregulation. And separately they are talking about crustaceans like prawns, and in these antennal glands or green glands are present. Since this is the list from NCERT, every student should compulsorily remember this particular list. Clear? Next one. Now we are talking about human excretory system. Please remember, once again I am telling you, we are studying only the NCERT content. We are not even going a single point out of NCERT. Clear? Now observe carefully. Human excretory system. What are present in the human excretory system? Two kidneys are present. That means a pair of kidneys are present. And then ureters, this is the one ureter, this is the other ureter. So total 2 ureters are present. That's why they are saying that one pair of ureters are present, and then one urinary bladder is present, and from the urinary bladder there is urethra. So one urethra is present. So number is also important. Pair means 2, 2 in number. So what are the structures that are present as pairs, and what are the structures that are present single? So this also you need to remember. Clear? So in this way you please remember. Now we will go with one after the other. First we will focus on the kidneys. Majority of the information they are talking is with respect to kidneys and its functional unit that is called as a nephron. Yes or no? That we are talking here. Regarding the kidneys, what is the color here? So they are dark brown color, and regarding the shape, they are bean shaped. Yes or no? So that's why generally we talk about bean shaped or kidney shaped, right? So kidneys are bean shaped. So that's why we are using these words. Okay. Next one, size. In NCERT they have given some values. So please remember that. And average weight is 120 to 170 grams for each kidney. Next one, these kidneys are covered with a connective tissue, and that connective tissue is called as capsule. It is called as capsule. And next one we are talking is regarding the regions. What are the regions that they are talking? Outer region of the kidney is called as cortex, and the inner portion is called as the medulla. Outer cortex, inner medulla. So these are the two important names that they are talking here. Next one they are talking is regarding the hilum. What is this hilum? If you're talking about the kidneys in the NCERT words, if you're observing, so these are the kidneys, right? These are the outer sides. Outer is a convex shape, outer convex, and the inner side they are having concave shape. And this inner concave notch, that notch is called as hilum. Please remember this. For one, if you are observing, this is the hilum. So this notch is called as the hilum. And at this hilum, what are present? At this hilum, see here, blood vessels are entering into this, and even the ureters are also present at this hilum. And not only this, even the nerves are also present at this hilum. So at this hilum, what are entering into this kidney? At the location of the hilum, blood vessels are entering, ureters are entering, and even the nerves are also entering into the kidney. This is the sentence very clearly given in NCERT. So please remember this. And this is about the hilum. And then they are talking about this particular kidney. In this kidney, what they are saying, outermost it is having a capsule. It is a fibrous connective tissue. And then inside this, for this kidney, if you are talking, outer portion of the kidney is called as the cortex, and the middle is called as the medulla. Yes or no? And this medulla, actually it is present like a cone shaped structures. Cone shaped structures or pyramid like structures. That's why they are called as medullary pyramids. These are called as medullary pyramids, and these pyramids they are opening into calyx. Singular is called as calyx. Likewise, many are there. For each one there is one. Likewise, so that's why plural sense if you are talking they are called as renal calyces. CALYCES, renal calyces. So medullary pyramids they are opening into renal calyx. Singular, plural if you are talking renal calyces. Now these renal calyces actually now they are opening into this funnel shape. Now it is called as a renal pelvis. So renal calyces they are opening into renal pelvis. Understood or not? And from this renal pelvis again, now this ureter will start. So in this way, now it is present. And then this outer cortex actually it is extending in between these medullary pyramids, and that's why they are called as the renal colons. This renal colon is actually cortex. Please remember this, cortex is extending between the medullary pyramid, and these extended portions of cortex are called as renal colons, and they are also called as colons of Bertini. Colons of Bertini. Understood or not? So colons of Bertini are actually part of cortex only, because they are saying that cortex is extended, right? So they are part of cortex. So please remember this is what the overall structure of this. And here whatever the red they have mentioned, this is the renal artery that is bringing oxygenated blood to the kidney, and this will be dividing and dividing. Finally it is branch, and one of its branch is afferent arteriole, that I will show you separately. So please remember this. And ultimately now they will utilize this oxygen, and then CO2 will be released into this, and once CO2 is released, now it will be collected by the veins, and ultimately it is the renal vein that is coming out. and now this renal vein ultimately it will be connected to inferior vena cava, so that deoxygenated blood will be again going to heart. So this is what happening. So in this renal artery, renal vein. So this here you can clearly observe this. Is it clear? So this is the inferior vena cava. So renal vein is coming and it is connected to inferior vena cava. Now it will take it to the heart. Is it clear? So this is what the basic structure of this particular kidney. And in these kidney, now here what we are saying, here is the cortex, here is the medulla, this is what we are saying. Now at these positions, actually in the cortex and nearer to the medulla. Nearer to medulla means juxta medullary. Juxta means near or nearby. So nearby medulla, now in this cortex portion, and here what are present, and these are the functional units of kidney. They are called as nephrons. Nephrons are present, and in this nephron generally when we are talking, so we are talking about two things. One is regarding the glomerulus. This is the tuft. Tuft means it is the network like blood capillaries that are produced from efferent arteriole. And what is this efferent arteriole? This efferent arteriole is actually the branch of renal artery. Renal artery is branching, it is branched for several times, and finally it is divided into efferent arteriole. This efferent arteriole, here it is forming a network like capillaries, and this network is called as glomerulus, and it is having glomerulus and renal tubule. Now in the renal tubule what are present? Bowman's capsule is present, proximal convoluted tubule is present, Hilney's loop is present, distal convoluted tubule is present. In this way, from many nephrons now they are connected to this collecting duct. Likewise, many collecting ducts they will combine, and ultimately they will go into this, through this medullary pyramid they will open into renal calyx. From there they are going to renal pelvis. From there the filtrate or the urine will go into this ureter, and from this ureter it will go into the urinary bladder, and through the urethra it will be released outside. This is what the flow and sequence is, and this sequence you need to remember very clearly. Is it clear? And this is what they are saying. So now we will go with part by part. So what are these parts? How exactly they are present? What is their role? Please remember, here there is a huge information that we can discuss, but we are restricting ourselves to NCERT content only. Once again I am telling you this. Clear? Information is very clear. Observe the diagrams very carefully. Observe the labeling so that you can easily understand all these things. Clear? Now we will go with one by one. First we are talking is regarding the Bowman's capsule, and here there is a glomerulus. This is the efferent arteriole that is entering into the glomerulus, so that now it is making a network of capillaries. This network of capillaries is called as glomerulus, and ultimately it is going out. It is going away. Out means it is moving away from the Bowman's capsule, and this away, the one that is going away is called as efferent arteriole. Spellings you need to observe very carefully. Here A, here it is E. A is going into the whereas E is coming out. These are the things that you need to observe very carefully. And the one that is coming out is called efferent arteriole. This efferent arteriole, now it is coming out, and now here around these tubules, now it is making a network of capillaries, and that's why it is called as peritubular network. It is called as peritubular network. Slowly I will tell you all these things, don't worry. This is what the information that they are talking here. Now here they are talking about another word. What is that word? Now observe carefully. So for this renal tubule is having Bowman's capsule, proximal convoluted tubule, Henle's loop, distal convoluted tubule. Yes or no? Now here what they are talking is this glomerulus plus this Bowman's capsule. Glomerulus plus Bowman's capsule. Now it is called as, they have given a different name. Glomerulus plus Bowman's capsule is called as Malpighian body. It is also called as Malpighian corpuscle. It is also called as renal corpuscle. Please remember these names. What is this renal corpuscle or Malpighian body? It is the glomerulus plus Bowman's capsule. Combinedly it is called as this particular renal corpuscle or Malpighian corpuscle or Malpighian body. Remember these names. And now this Bowman's capsule, inside it is having a space, lumen. That lumen is continuous. That lumen is continuous like this. Understood or not? Now slowly we will understand all these things. Now here they are talking about these regions. So observe very carefully. Quickly I will tell you about all these things in a rough diagram. I am drawing only just I am drawing the rough diagrams. Observe carefully. Now Bowman's capsule is present like this. Understood or not? It is a bowl-like structure. Clear? Now in this what happens? Now efferent arteriole is entering into this, whereas efferent arteriole is going out. Understood or not? And here it is making a big network here. Here it is making a network of capillaries. Understood? And this network of capillaries is called as glomerulus. Glomerulus. Observe carefully. And then now this is the Bowman's capsule. And inside this it is having a space, and this space is called as lumen. Is a general term, right? And now this is continuous with a highly folded structure. This is called as proximal convoluted tubule. Proximal means nearer. Nearer to what? It is nearer to this Bowman's capsule. Structurally, it is continuation with Bowman's capsule. That's why it is proximal. Proximal means nearer. Convoluted means highly folded tubule. This is the proximal convoluted tubule. And now this proximal convoluted tubule is continuous with Henle's loop. It is called as Henle's loop. It is a U-shaped structure. U-shaped structure. And please remember, diagrammatically there is a purpose here. That's why I am telling you here. And this is the descending limb. This is the descending limb of Henle. This is going down, right? So that's why it is descending loop of Henle. Descending limb of loop of Henle. And generally it is a thin. And now on the other side it is going up. It is going up. That's why it is called as ascending limb of loop of Henle. Ascending limb of loop of Henle. In this also, the initial portion of ascending limb is thin, and then it is thick, and then it is thick. Observe carefully. Okay. And then finally what happens? Here also it is again continuation with, again there is a folded structure, and this is called as distal convoluted tubule. And structurally this is present away from this Bowman's capsule. So structural connection, if you are observing, this is also convoluted, this is also convoluted, but this is nearer to Bowman's capsule, this is present far away from Bowman's capsule. That's why this is a proximal convoluted tubule, this is distal convoluted tubule. And this distal convoluted tubule, now it is opening into a collecting duct. This is the collecting duct. Now in this way many nephrons will open into this collecting duct. So this collecting duct is collecting the urine, and that urine is passing through these collecting ducts. So now where these collecting ducts are present? So it actually started in the cortex. Now it is passing through medulla, and then through the medullary pyramids. Medullary pyramids, it is entering into the calyx, then into the renal pelvis, then into the ureter. Then the urine will go into this urinary bladder. So this is what the flow of urine in these particular tubes. Is it clear? This is the general basic idea regarding what are the various parts that are present within this nephron. Clear? So this is what we are talking. And here we have completed this glomerulus, and also we have completed the renal tubule. Basic structures. One by one we will study. First we studied all these names. Clear? With this part is completed. This information also we have completed. Then we are talking about the nephrons. We are talking about two types of nephrons based on their location. So what is this particular piece of information is? So here for the, suppose this is the outer cortex and this is the inner medulla. Inner medulla. Here what they are saying is there are some nephrons here. There are some nephrons here. So here what happens is they are present like this, and here now they are opening into this collecting duct. This is the collecting duct, right? Collecting duct is started in the cortex. It is moving in the medulla. From this medulla now it will enter into the renal calyx. So in this way it is keep on going. Okay. Suppose you are observing this as a conical structure. This is how it is present. Understood or not? So this is how it will open into this renal calyx. Okay, the thing is, there are some nephrons that are present within the cortex region only. But for these nephrons, only a small portion of Henle's lobe, is coming into this medullary region. Whereas There are some other types of nephrons. There are some other types of nephrons. For these nephrons also, Bowman's capsule and proximal convoluted tubule are present in the cortex. But these nephrons are having very long Henle loop, very long Henle loop. And for these, if you are observing, major portion of Henle loop is present within the medullary portion. Whereas Bowman's capsule, proximal convoluted tubule, distal convoluted tubule, all these are present within the cortex region. In this way also some nephrons are present. Please observe carefully. Two types of nephrons are present. Majority of these nephrons are present within the cortex region, and there are some nephrons that are present very nearer to, very close to medullary portion. And for these nephrons, Henle's loop is very, very long, and majority of the portion of Henle's loop is present within the medullary portion. Whereas those nephrons which are mainly present in cortex, they are having very short Henle loop, and the portion of Henle loop present within the medulla is very, very less. In this way, there are two types of nephrons, and this type of nephrons are called as cortical nephrons. Cortical nephrons. And these nephrons are present very close to medulla. That's why these are called as juxta. Juxta means nearer. Nearer to what? Medulla. So that's why these are called as juxtamedullary nephrons. So cortical nephrons, juxtamedullary nephrons. So in this way, they are talking about two types of nephrons. So this is the cortical nephron, but this is the juxtamedullary nephron. So please observe these diagrams very carefully. Very carefully. Don't make any mistakes. This is what they are talking. Clear? These names also clear have mentioned, and this is what the explanation for this. Clear? Next one. Peritubular network. Who is forming this peritubular network? See here. Efferent arteriole is entering into this Bowman's capsule. Okay, that means the cup-like structure, the space the formed by this Bowman's capsule. So into that efferent is going, and there it is making a network of capillaries called as glomerulus, and the remaining blood vessel is coming out. The one that is coming out is called as efferent, efferent arteriole. And please observe carefully, this efferent arteriole is having a branching network of capillaries around these tubules, proximal, distal and Henle's loop. Around all these things, it is making a network like structure. This is called as peritubular network. Peri means around, tubules. These are the tubules, right? Around these tubules, now it is making a network. That's why it is called a peritubular network. In this peritubular network, there is a vessel that is moving almost parallel to Henle's loop. There is a vessel that is moving almost parallel to Henle's loop, and it is called as vasa recta. Very, very important for examination. So please remember this. This is called as vasa recta. Clear? Regarding the vasa recta, in the next coming sections, we will discuss more. So please remember this. This is what about the peritubular network. This is what about the general basic information regarding the nephron. Clear? With this, the basic information regarding the kidney and the basic information regarding the nephron, we have completed. Now we are going further, and here we are talking about how exactly the urine will be formed. For the urine formation, there are actually three important steps. There are three important steps. What are these steps? First one is glomerular filtration. It is also called as ultrafiltration. Next one is reabsorption and secretion. Please remember very carefully, read the NCERT very carefully. You will find some words called selective reabsorption, selective secretion. Why they are using these words? Because depending on the condition, these two things are happening. So that's why they are technically using this. But anyhow, when I am giving explanation for this, I will specifically inform you regarding these things. Okay. Sorry. First, let us focus on this glomerular filtration. What is this glomerular filtration actually? So the thing is why it should happen. First question is why it should happen, and where it is happening, and how exactly it is happening. These are the things that we need to understand very clearly. Now let us observe carefully. So whatever the food that we are eating, that is going through a tube-like structure, this is called as digestive tract. In this digestive tract, there is a portion called as intestine. So now this food will be digested. It will be broken into small components, so that now it will be absorbed into the blood. it will be absorbed into the blood. Already I told you regarding that portal system, right? From the intestine, there is a vein that is called as hepatic portal vein. All these are called as veins only, no arteries. Okay, hepatic portal vein. And this hepatic portal vein, it will take it to the liver. This is the liver. Understood or not? So now what happens? Now liver will utilize these food materials. In these food materials, mainly we are talking about the proteins, amino acids, NH2. So it is having NH2, C, and this is H, and this is COOH, and this is the R group. This is amino acid. In this amino acid, here technically it is having one amino group. So in majority of the cases, when it is excess, now this amino group will be released. And this amino group along with CO2, it is forming urea. It is forming urea in the case of human. And now this urea will be released into the hepatic vein. It is the vein that is coming from the liver. So in the hepatic vein, if you technically see, hepatic vein will have more and more concentration of urea. Now this hepatic vein from this will go to the heart. It is going to the heart. Yes or no? Deoxygenated blood, right? It is going to the heart. And from this heart, now what happens? Now it is going to the lungs for oxygenation. And then the oxygenated blood is coming back to the heart. And from this heart, now it is going to these kidneys. From this heart, it is going to the kidneys. And what is that blood vessel that is taking this? Renal artery. Renal artery is dividing and dividing, and finally it is forming arteriole. It is forming arteriole. Observe carefully. It is the efferent arteriole. It is the efferent arteriole. Efferent arteriole. That means this efferent arteriole has nitrogenous waste materials like urea. I am talking only for urea, but there are many other types. For easy understanding, I am talking with respect to urea. So efferent arteriole is having more urea. Yes or no? Now remember this. Now it is having nitrogenous waste materials. Now this efferent arteriole is having oxygenated blood, but it is having more and more nitrogenous waste materials. So now it is coming here. Now what should happen? Now here it should, now it is a network here. Yes or no? Capillary network. Now here what should happen? Now technically, direct point if you want to talk. So what should happen here? Nitrogenous waste from this blood vessel should enter into this particular one. Yes or no? So please remember, entirely it is not possible to remove this. Major quantity will be removed from this, and the remaining is going here. And this is the efferent arteriole. So there is a chance that they may ask a question. One is efferent arteriole, other one is efferent arteriole. Where you can find more concentration of urea or nitrogenous waste? In the efferent arteriole or in the efferent arteriole? Definitely in the efferent arteriole, because efferent arteriole, now the blood from this is coming into this glomerulus. In the glomerulus, filtration is taking place. And what is the main aim of the filtration? Removal of this waste from the blood, removal of the waste from the blood, so that now this waste material called urea or any other nitrogenous waste is going into the Bowman's capsule, lumen of Bowman's capsule, so that efferent arteriole is having less concentration. In this way, the nitrogenous waste produced by the liver is removed by the kidneys. This is what the ultimate conclusion that you need to understand. Then how exactly it is removing? Now we will talk about that filtration process. Now here what happens is, this is the capillary. So capillary means it will have the endothelium. Capillary is having endothelium. Already in that body fluids and circulation we studied, right? So endothelium is nothing but simple squamous epithelium. Now it is having a very thin diameter, right? So thickness is very less. So that's why now the molecules can easily pass through this, and there are some small gaps that are present here, so that now water and small molecules can easily pass through this. This is what for the capillary. And now this is the Bowman's capsule. So for this Bowman's capsule, now it is having some cells, and these cells are called as podocytes. These are called as podocytes. This is the epithelium of Bowman's capsule. Epithelium of Bowman's capsule is having some cells, and these cells are called as podocytes. Yes or no? And in these podocytes, here there are some small slits are there, and these are called as slit pores. Slit pores. Understood or not? Slit pores are present, small gaps are present. And what is the advantage of these gaps? The advantage of these gaps is now they will allow the movement of water, small molecules, ions from the blood into the lumen of Bowman's capsule. This is what the advantage is. So here there are two layers. One layer is, it is the endothelium of capillary blood wall, and then other one is the epithelium of Bowman's capsule. These are the two layers, and in between these two, there is a layer, that is called as basement membrane. There is a layer called as basement membrane that will provide support between these. So in this way, total 3 layers are present here. So one is the endothelium of capillary blood vessel, and then other one is the epithelium of Bowman's capsule that is having the cells called podocytes, and in between these two layers there is a basement membrane. So in this way, total 3 layers are present at this filtration location, Understood or not? And here please remember, this basement membrane is actually having some fibers. Again, it is a network like structure. That means it is also having some pores, and here it is very thin, and here some substance can pass through like this. Now what are coming here? Water is coming here, and then some minerals are coming here, and then nutrients are also coming into this. Nutrients like glucose, amino acids, many things are coming into this. Please understand this. So in this way they are coming through this. So in this way some filtration is taking place. So filtration is taking place means something is left over here. What is left within this blood? All the blood cells are left here. Please remember here, blood cells are not passing through this. Blood cells are left within this blood vessel itself. Not only blood vessels. Now here there are some proteins. Proteins are present within this particular blood. Please remember this. Cells are not coming here, proteins are not coming here. These two are present here. And many students are thinking that, what many students are thinking is, entire water is coming into this. No, if entire water is coming here, how it will flow here? Some amount of water is present. Please remember, this is what I am saying, that don't think that entire water, mineral, nutrients, everything are coming here. No. Whatever they are coming are water, minerals, nutrients, all these are coming, but still some amount of water and some things are present here. That's why it is able to flow. Right? If entire water is coming, how only cells and proteins will move there? Water should be there. It is a medium, right, for the transport of all these things. So majority of the content is coming here, but still a small quantity is left over here. So that's why that leftover, whatever the fluid is there, that fluid means plasma, along with all these things, now it is passing in this efferent arteriole. Please understand this. This is what happening. So at this glomerulus and at the Bowman's capsule, here actually the filtration is taking place, and this filtration is called as glomerular filtration. And here, since they are very fine gaps are present, slits are present, through this filtration is taking place. It is also called as ultrafiltration. In this way, the filtrate, please understand these technical names. Filtrate. In this way, upon filtration, the fluid coming into the lumen of Bowman's capsule is called as filtrate. And what are present in this filtrate? Water is present, minerals are present, nutrients are present, and along with this, what are these? Nitrogenous waste materials. This is the most important one, right? Nitrogenous wastes are present. Main aim is to remove nitrogenous waste only, right? So now all these are present here. And this filtrate, initial filtrate, now it is passing through this lumen. So initially it is present in Bowman's capsule, then it will pass through proximal convoluted tubule, then it will pass through Henle's loop, then it will pass through distal convoluted tubule, and then it will go into the collecting duct. But this initial filtrate is not as it is going there. When it is passing through these things, next steps will come. What are the next steps here? We studied only the first thing, right? What is the first thing? It is only glomerular filtration we studied. And before it is converted into urine, there are some things that are happening. What is happening here? Reabsorption is taking place, secretion is taking place at various locations. Proximal convoluted tubule, Henle loop, distal convoluted tubule, and collecting duct, even in the collecting duct also. In all these locations, reabsorptions are taking place, and then secretions are also taking place. And finally, whatever is left over, after doing all these things, whatever is formed, that will be called as urine. Please understand this. So in this way, reabsorption and secretion. So we have completed this glomerular filtration, and now we need to focus on reabsorption and secretion. Before going to that, now let us see this diagram. So this is the Malpighian corpuscle, and this is the Bowman's capsule, and this is the efferent arteriole. This is the network that is formed, and this is the efferent arteriole that is going out here. Now here at this junction, you can see that this is the endothelium of capillary blood vessel, and they are fenestrated. They are having some small gaps, and here in between this there is a basement membrane. And then here this is the epithelium of Bowman's capsule, and these cells are called as podocyte cells. So in this way, total 3 layers are there. Now through these gaps, now filtration will occur, and that's why this is the filtration membrane we are observing here. Understood or not? This is what they are saying, but you don't need much technical details, but at least basic information, basic points you need to understand for examination. Clear? So that you should not face any problem here. And here they are talking about GFR. What is this GFR? Glomerular filtration rate. So here it is undergoing the filtration. On an average, if you are talking with respect to numerical values, what they are saying, heart in 1 minute from each ventricle. Please remember heart calculation, that cardiac output we have calculated from each ventricle we are talking, right? So what is the value that we are talking? Approximately 5 liters of blood is pumped by each ventricle in 1 minute. But here, how much of blood is coming to this kidneys for the filtration process here? Approximately 1100 to 1200 ml. 1100 to 1200 ml of blood is coming to these kidneys for filtration process or purification process. That means it is approximately 1/5 of the blood that is pumped by each ventricle. Yes or no? So that much of blood is coming here, and out of this blood, upon filtration, how much it is forming here? 125 ml per minute it is forming. All the calculations we are talking for per minute only, so please remember here. So approximately 1100 to 1200 blood is coming to the kidneys, but out of that, how much filtrate that is formed? Just it is 125 ml per minute. That means you can understand that you can understand whatever the point I said now here. Entire fluid is not coming here. Some volume of it is undergoing filtered here, remaining is going like this. Is it clear? So please understand this. So in this, and this calculation is for both the kidneys. So 125 ml per minute is produced. That means for 1 day, if you are calculating, how you will calculate for 1 day? For 1 minute it is 125 ml. For 1 hour it will be, how you will do? 125 ml into 60. That will be for 1 hour. 1 day means 24. So again multiply with 24. So 125 ml into 60 into 24. Do it. You will get 1,80,000. 1,80,000 milliliters per day. 1,80,000 milliliters per day. You just convert into liters. What you will do? Divide with 1000. So you will get 180 liters per day. This is what the original calculation is. This is how you need to calculate this. So in this way, this much is produced. This is the filtrate that is produced. But do you think that this much of urine is produced? No. This is the initial filtrate that is produced per day. Okay. But what is the ultimately urine that is produced? It is almost 1 liter to 1.5 liter per day. That's it. That means approximately 99% of this is again reabsorbed. Now you can understand what is the role of that reabsorption. If that reabsorption is not taking place, what happens? You need to continuously drink the water. That's it. Yes or no? This is what the problem is. So understand these values, interlink with one paragraph with other paragraph, then you will understand the concept carefully. So that's why compulsorily there is a requirement for reabsorption, and not just reabsorption, even some other ions are also, even some waste metals are also released, secreted by the cells of the nephron also. Yes or no? So from the kidney also some waste materials will be produced, they also should be released. So now they also will be secreted. So that's why reabsorption and secretion is also very, very important point. And before directly going into that, in the NCRT they are talking about juxtaglomerular apparatus. Just now I told you the meaning of juxta. What is the meaning of juxta? Nearby. Nearer to what? Glomerulus. Nearer to the glomerulus, you can see some apparatus, some arrangement we are observing. What is that? This is the glomerulus. Yes or no? And this is efferent arteriole, efferent arteriole, and this is the efferent arteriole. Clear? And here this is the distal convoluted tubule, DCT, distal convoluted tubule. Now observe carefully, distal convoluted tubule. For this, some cells of this, some portion of this distal convoluted tubule is present very close to this efferent and efferent arterioles. At those locations, these cells are little bit larger in size. And I am not going into those names now because they have not mentioned in NCRT, but in the regular class we have discussed about all these things. Clear? And now this particular apparatus is called as juxtaglomerular apparatus. Clear? This is what. And these cells, the cells of this, they are secreting a hormone that is called as renin. And this renin hormone is very, very important. Now let us quickly read the NCRT sentence. What they are saying here, that renin, anyhow at the end of the chapter we will study renin angiotensin mechanism. That renin will convert angiotensinogen into angiotensin 1, and that is further converted into angiotensin 2, and that angiotensin 2 ultimately helps in increasing the glomerular filtration rate, GFR. This is what the ultimate conclusion they gave. Now let us see. A fall in GFR, if GFR is decreased, then what happens? It can activate the juxtaglomerular cells. It is activating the cells of juxtaglomerular apparatus to release renin, R-E-N-I-N, renin. Spelling is very, very important. R-E-N-I-N, renin is the hormone that is released by juxtaglomerular apparatus. But there is another spelling. R-E-N-N-I-N. R-E-N-N-I-N is the enzyme that is involving in digestion of milk protein in the infants, and that too its action is in the stomach. So that is actually related to digestion and absorption chapter, but anyhow that chapter is deleted, so leave it. And the spelling is very important. It is R-E-N-I-N, renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. So when GFR is decreased, now juxtaglomerular apparatus cells, they are releasing renin. So ultimately it is increasing the GFR so that it will come back to normal. This is what the ultimate role is, and regarding this, anyhow at the end of this chapter we will study more. Clear? Now let us see about the other things. See, under the urine formation, three things we are talking. First one is glomerular filtration, also called as ultrafiltration. Next two things are reabsorption and secretion. And these things you can easily understand from this NCRT diagram. What is this reabsorption? From which locations these reabsorptions are taking place, and those locations, what are the components that they are secreting? Now observe very carefully. Glomerular filtration is occurring here. Yes or no? Upon filtration, the filtrate is coming into the lumen of Bowman's capsule. Then it is passing through the lumen of this nephron. Yes or no? It is passing through like this. Now what you are saying, after this Bowman's capsule, now the filtrate is coming into this proximal convoluted tubule. Yes or no? And this proximal, please remember this one, this upper part, these parts are present within the cortex, and this part is present within the medulla. These portions also you need to remember. Now I don't need to mention, but please remember this. Now this proximal convoluted tubule, now observe this. These upper arrow marks are reabsorption. They are absorbing from the filtrate. This is the filtrate, right? From the filtrate, they are absorbing into the surrounding cells. Please observe this carefully. They are absorbing. Now, what are these things? So, these arrow marks are going into the tube. These arrow marks are going into the tube means these cells, the cells present in this location, they are secreting these into the tube. So, please observe the arrow marks carefully. Understood or not? So, here, so in this particular portion, these arrow marks present on the upper side, these are all related to reabsorption. That means from this filtrate, what are reabsorbed? Bicarbonate ion is reabsorbed, NaCl is reabsorbed, water is reabsorbed, nutrients are reabsorbed, K plus is reabsorbed. And what are released into the filtrate? H plus is released and ammonia is released. Anyhow, ammonia is the nitrogenous waste, so it is released. And why H plus is released? Please remember here. These reabsorptions and secretions are a selective process. What is the meaning of this selective? It all depends on the body conditions. Please remember this. What is the meaning of this selective secretion, selective reabsorption? Based on the body conditions. If something is required for the body, then it will be reabsorbed. If it is not required, now it will be released, means secreted. So always it is depending on the body conditions at that particular time. So that's why sometimes some ions may be absorbed, sometimes the same ions may be secreted, because it all depends on the body conditions. At that particular situation, is it required or not? That condition will decide whether it should be reabsorbed or it should be secreted. So please remember, even though they have given like this, it all depends on the body conditions. According to those particular situations, it will be decided whether it has to be absorbed or it has to be secreted. So that's why read the NCERT sentences carefully. Still, in general, they are saying that at these locations, these are reabsorbed, these are secreted, but it all depends on the conditions. Clear? So this is what they are saying. And that too, if you are clearly reading the NCERT, what they are saying, 70 to 80%, 70 to 80% of the reabsorption, that means maximum reabsorption is taking place in proximal convoluted tubule. So that's why majority of the components are again absorbed back in proximal convoluted tubule itself. So in the remaining portions, the reabsorption is all based on the body requirements. Anyhow, majority of the contents are going here, so it is compulsory. So 70 to 80% are absorbed here. For the regarding the remaining contents, it all depends on the body conditions. Based on this, at different locations, again different contents are reabsorbed and different contents are secreted. So please read the NCERT, keep this diagram and then observe the NCERT sentence every time. Then you can understand the NCERT sentences. This is for the proximal convoluted tubule. Then the filtrate is keep on moving into this Henle's loop. In the Henle's loop, this descending loop is thin. Please remember, descending loop is thin. And in this thin descending loop, what happens is water will be reabsorbed. So this descending loop is permeable to water, but the reabsorption of these ions like Na plus Cl minus K plus, all these things are very rare, very rare. So that's why they say that it is less permeable to ions, but it is more permeable to water. That's why water can be easily absorbed in the descending loop of Henle. Remember this content. I will link up this with concentration content. Okay, urine concentration mechanism, that counter current mechanism is there, no? That I will link up. So please remember this. Next one, Henle loop, again turning back and it is moving up here. And this part is completely called as ascending limb. In the ascending limb also, the initial portion is thin. That's why it is called thin segment of ascending limb. Then it is thick. So it is the thick segment of ascending limb. So please remember, this ascending limb is impermeable to water. Descending is permeable to water, but ascending is impermeable to water. Opposite. Observe carefully. And for descending limb, what they are saying, it is almost impermeable to ions. But here ions will be reabsorbed either passively or actively. Passively means without the use of energy. Actively means by using energy. So now it will be happening here. So that's why NaCl is absorbed here. Observe carefully. This is what happening here. Clear? And not only this, urea is released into the tubule here. Urea is released here, that means it is secreted. So if it is excess, please remember why urea is present here. Actually urea is a nitrogenous waste, but still some amount of urea is present within the medulla, because NaCl and urea, they are trying to maintain higher concentrations. I will show you that. Mechanism and everything. So in this way, some amount of urea is required here. If more and more urea is coming from collecting duct, now what happens? Excess should be released. Now that excess urea is once again released into this ascending limb, so that now it can be released out. So in this way, these water here and ions here, now they are absorbed, reabsorbed, and urea is secreted into the ascending limb. Understand all these things. Observe the diagram carefully, you will get this. Now come to this distal convoluted tubule. In the distal convoluted tubule, upper these things, NaCl, water, bicarbonate, all these can be reabsorbed based on conditions, requirements. If it is required, now they will be reabsorbed. Next one, K plus and H plus, now they will be secreted again to maintain acid base balance, again to maintain Na plus K plus balance. If H plus ion concentration is more, now it will be released, secreted. If it is not more, if it is less, then it is not secreted. It will take up here and then it will maintain here. So in this way, all these are to maintain as per the required conditions. Please remember this. That's why all these are selective processes. That's why they are not always happening. So based on the body conditions, they are happening. Clear? And then now the remaining filtrate is coming into this collecting duct. And from the collecting duct here you can use the term called as urine. Yes or no? Once it is crossing this distal convoluted tubule and once this filtrate is coming into this collecting duct, then you can use the term called as urine. So that's why in the NCERT they said that the urine will be 4 times concentrated. That also I will tell you under a separate diagram. Don't worry. So this is what about that glomerular filtration, selective reabsorption, and then selective secretion. These are the 3 things. So because of this, after all these things, whatever is left over, that is present here, and now it is keep on moving, and that you can call it as urine. So that's why for urine formation you are studying about all these 3 steps. Is it clear? This is what the relation that you need to understand. Once it is completed, now functions of the tubules, just now we have studied, at the same time we have studied. So some are involving in absorption of some things and secretion of some things. In this way, different regions are having different functions. This is what we are saying. And we have completed up to the DCT, and now we will focus on the collecting duct. And from the collecting duct also, water will be reabsorbed. Water will be absorbed by medullary interstitium, and a small amount of urea is reabsorbed to maintain the concentration or the osmolarity of the medullary interstitium. These are reabsorbed, and depending on the condition, NaH plus, K plus, they also will be secreted. Read the NCERT content. They have given that even some ions are also secreted into this particular filtrate at this collecting duct portion. This is also there. So these are all based on body requirements and the conditions. So this is what the information regarding the functions of these particular tubules. And after completion of this, now we are talking about the mechanism of concentration of the filtrate, how this filtrate is getting concentrated. Clear? Now here if you are observing, now from this portion onwards, this is the cortex, this is the medulla. In the medulla also, this is the outer medulla, this is inner medulla. And now observe carefully. In the cortex, what is the concentration that you are observing? It is 300 milliosmoles per liter. And when you are observing for outer medulla, now you just observe it. So here it is coming around 600 milliosmoles per liter. When you are talking about this inner medulla, now it is 900 milliosmoles. And here at this U-shaped curve of Henle loop, here you can observe approximately 1200 milliosmoles per liter. So here it is 300, but here it is 1200. That means how many times it is more concentrated? 4 times more concentrated. So the filtrate that is present here is having some concentration of 300 milliosmoles, and when it is coming here, it will become 1200 milliosmoles. So 300 to 1200 means how many times it is concentrated? It will be 4 times concentrated, very clearly mentioned in NCERT. So please observe carefully. Clear? But the thing is, immediately after glomerular filtration, it is 300 milliosmoles per liter. Then here it is increasing. When it is coming down the Henle loop, descending loop, the concentration is increasing. Again in the ascending loop, it is decreasing. Why it has to increase, why it has to decrease, and again why it has to increase here? What is the role of this? Why it is doing? And whatever is absorbed, where it is going? These are the points that you need to observe carefully. Understood or not? So for these things, I will draw the rough diagram, and in the diagram I will explain you, because here they gave separately. This is the loop of Henle, this is the vasa recta they gave. So at this much of distance you cannot understand. So I will draw a rough diagram, don't worry. So here what are the major important points? Before understanding this, many students, they are trying to by heart this, okay. But if you don't understand the logic, there is no use in studying this. So first I will tell you the logic, why you need to study this. Why this reabsorption concentration are very, very important. Now I will tell you in simple. I will draw different diagrams, don't worry. So observe carefully here. What actually we are saying is, this is the Bowman's capsule and this is the This is the proximal convoluted tubule, and this is the Henle's loop, and this is a part of Henle's loop. Okay, and then this is the this is the distal convoluted tubule, and this is attached to collecting duct. Understood or not? Here what exactly we are saying is, this is the efferent arteriole, and then this is the efferent arteriole, and here it is forming the network that is called as glomerulus, and efferent arteriole is forming peritubular network. And for your understanding purpose, I am drawing the diagram like this. Observe carefully. So this is the vasa recta directly I am drawing. Anyhow, peritubular network is present, and directly the vasa recta I am drawing here. This is what happening, right? Now observe carefully why this reabsorption is very important. Now understand the logic here. Why reabsorption is important? Because actually blood is coming or entering into the nephron through this efferent arteriole. Very good. And when it is coming, it is having some volume. And what is the main purpose of entering into this particular one? The main purpose is to remove the nitrogenous waste materials. Yes or no? This is what the ultimate aim is. So nitrogenous waste should be removed from this. But the problem is, see, our ultimate aim is, from this blood only nitrogenous waste should be removed. If it is happening, then it is fine, no problem. But actually what is happening, it is not just the nitrogenous waste. Water is also going, minerals are also going, and then nutrients are also going into this particular tube. Yes or no? So here you can observe two things. What are the two things? The volume of blood that is entering into the glomerulus is not same as that of volume of blood that is going out. Do you able to understand or not? I will repeat once again. The volume of blood that is coming into the glomerulus is more when compared with volume of blood that is leaving the glomerulus. Volume of blood that is leaving the glomerulus is definitely less, because see, much fluid is coming into this. So here the volume will decrease. Volume will decrease. So when this volume is decreasing, now it is keep on going, and ultimately it is going to the heart itself. And finally, what is the overall value of the volume? It will be decreased. Please remember here, if it is not going back, please observe carefully. If this volume is not going back, what happens? In every filtration, what happens? The blood volume is keep on decreasing. That is dangerous. That is dangerous. So this blood volume should be maintained. So how you will maintain this? Now observe the logic. So first thing is, we need to maintain the blood volume. This is the first point you need to understand. Second point is, not only nitrogenous waste, water, minerals and many other things, they are present in this. If they are not reabsorbed, what happens? Now they will be released out along with the urine. That means every time you need to eat and you need to take all these water, minerals and all these things. And that is not possible. Understood or not? So two things are there. One is fluid volume is getting decreased. Second thing is, if this is not reabsorbed, every time it will be released out. So two things are there. So both the things we are solving at the same time. Both the things we are solving at the same time. How we are solving? First I will give you summary, then we will go with mechanism. What is the overall conclusion here? So okay, here along with the nitrogenous waste, water, minerals and many other things are coming here. So now what should happen? From this particular lumen, leave the nitrogenous waste. Keep the nitrogenous waste in the lumen itself. Water, minerals, other things, let it absorb into the interstitial cells. This is the medullary cells, right? Medullary interstitial cells. First they will absorb. And from this, now all these things, they will enter into this particular efferent. This is all efferent, right? So in this efferent, now they are entering. Now what is the ultimate meaning? Now once again the original blood volume is maintained. Original blood volume is maintained here. And then nitrogenous waste is removed along with some small volume of water. That's it. Now both the problems are solved. How both the problems are solved? Now this whatever the volume that is going here, majority of the volume is coming back into the blood vessel again. This is the blood vessel again. That means blood volume will be maintained. Yes or no? Second point. Now nitrogenous waste is removed. So nitrogenous waste has to be removed. and that purpose is also solved, and this nitrogenous waste is removed. So both of them are done at the same time, and for this we need to talk about this concentration of the urine mechanism, how exactly it is concentrated. And here we are talking about another word that is called as counter current mechanism. Counter current mechanism. So many students are not understanding what actually the counter current is. Counter current. What is the meaning of counter current? Simply, what is the meaning of counter current? Movement in opposite direction is called counter current. Counter opposite current movement. Counter is opposite, current is the movement. Counter current is the opposite movement. Movement in opposite directions. Movement in opposite directions is called as counter current. That's it. Here also you are studying something that is moving in opposite directions. That's why here also you are using a term called counter current. If you understand this opposite movement as counter current, you understood 50% of this. Now you need to focus on then where exactly they are moving in opposite direction. Now this is a very good question. Now let us see, this is the Bowman's capsule, proximal convoluted tubule, this is the Henle loop, this is the Henle loop, and this is the distal convoluted tubule. Now it is opening into the collecting duct. Understood or not? Now we will see. I will use different colors to show opposite directions. Observe carefully. Now the filtrate is moving in the descending limb of Henle. In the descending limb of Henle loop, this is the direction of filtrate movement. But in the ascending limb of Henle, this is the movement of filtrate. Now in these two limbs, the filtrate is moving in opposite directions. Yes or no? Here it is moving in opposite direction. So this is called as one counter current. This is called as one counter current. Observe carefully. Next one. Second counter current. The movement of filtrate in ascending limb is like this, whereas the movement of filtrate or urine in collecting duct is like this. So now these two are in opposite direction. This is another counter current. Observe all these things carefully. This is one counter current, this is another counter current, and there is third counter current. Where is the third one? Where is the third one? Now observe carefully. This is the efferent arteriole, and this is the efferent arteriole, and this is the glomerular network. Now this is the vasa recta. Directly I am drawing the vasa recta. Observe carefully. This is the vasa recta. This is the vasa recta that is present like this. Yes or no? Now observe carefully. Inside the vasa recta also, for the vasa recta, the movement of this blood is like this here, and here the movement is like this here. Now these two are in opposite direction. Yes or no? So that is the reason why total 3 counter currents we are observing. Where we are observing? First counter current we are observing where? In the descending limb and ascending limb of Henle loop. Second one, ascending limb of Henle loop and collecting duct. And the third one, descending limb of vasa recta and ascending limb of vasa recta. These are the 3 counter currents that we are observing. I hope now you understood what is the meaning of counter current. It is the movement of fluid in opposite directions here. This is called as counter current. So with respect to this we are observing 2, with respect to vasa recta we are observing 1, total 3 counter currents we are observing here. Clear? This is what about the counter current term and its meaning. But how exactly the concentration of urine is taking place? This you need to understand very carefully. But before directly going with that, first you need to understand some technical meanings. Please understand this. Some students are trying to by heart this. Don't try to by heart this. I will explain you the mechanism with clarity. Try to understand this. Okay. First now we are studying about some chemistry principles of chemistry. So with respect to solutions chapter. Okay. Now we will see this. Now you want to increase concentration of a solution. Please remember this. First thing is, I want to increase the concentration of solution. How I can increase the concentration of a solution? Think. Next one, second option. I want to decrease, this symbol is decrease symbol. I want to decrease concentration of a solution. If you understand these two, you can easily understand that urine concentration mechanism. So please understand this. If you understand this, in single minute you can understand the entire mechanism given in NCERT. So observe carefully. Now here, to increase concentration of a solution, we have two options. To increase concentration of solution, we have two options. Same way, to decrease concentration of a solution also we are having two options. What are these options? Now understand these options. How you can increase concentration of a solution? Solution has two things. One is a solute, other one is a solvent. Once again I am repeating, solution has solute and a solvent. I am talking about these two words. Now, how you can increase concentration of a solution? By adding solute. By adding solute. Yes or no? By adding solute, can you increase concentration of a solution? Yes, you can increase. Or in a different way, with respect to solvent, you just tell me. How you can increase the concentration of a solution? By removing solvent. Removing solvent also you can do this. Yes or no? Keep the solute like that and remove the solvent. Then also concentration will increase. Yes or no? Please understand these basic principles. Then only you can understand this mechanism. Concentration of a solution can be increased by adding solute or by removing the solvent. Yes or no? You can do this or you can do this. Both also you can do, but any one only we are focusing. Next one. Now here, how we can decrease concentration of solution? Exactly opposite. How you can decrease the concentration with respect to solute? By removing solute. By removing solute, you can decrease the concentration. That means if solute is removed, concentration will decrease. Next one, with respect to solvent, what you will do? You just add the solvent. Add the solvent. Add the solvent. Then concentration will decrease. Clear? Now in this case, what is our solvent? The solvent here is water. In our case, the solvent is water. Solvent is water. What are the solutes that we are talking here? Na plus, Cl minus, K plus, all these things. Here also the same. Understood or not? These are the things. If you understand this basic mechanism, you can easily understand what actually they are talking in the concentration of urine. So please observe this particular one. So here they have given a particular diagram, 300 milliosmoles, 600, 900, 1200. That means now here from in this diagram, if you are observing from top to bottom, the concentration is increasing. And the thing is, even inside the tubes also, the concentration should be maintained. Because outside these tubes also interstitial cells are present. In the medulla there are many cells, and in those cells this concentration is maintained. If this concentration is maintained in those cells, means that concentration will be exerted on these tubes also. So the solvent or whatever the solution that is present inside this, we can call it as a filtrate. The filtrate present in this or the blood present in this also should experience the same concentration. So that's why how it will be maintained. Now we will see this. For this, with a rough diagram I will try to explain you. So please listen very carefully, very carefully. Okay. Now let us start with Bowman's capsule. This is the Bowman's capsule. This is the proximal convoluted tubule. This is the proximal convoluted tubule. And this is the Henle's loop. I am making little bit wider because I need to draw vasa recta here. That's why I am making it a wider. Observe carefully. Okay. So now it is like this. Now it is connected with collecting duct. This is the collecting duct, distal convoluted tubule. This is the in the ascending limb also. So here it is thin and thick, likewise it is there, no? Yes, thin, thick, likewise. Let us make the difference. This is the ascending limb of Henle loop. Okay, this is the descending limb of Henle loop. Clear? This is the proximal convoluted tubule. This is the Bowman's capsule. Clear? Next one. Next one, since it is a blood vessel, I am drawing with red. Now let us observe carefully. Now this is the efferent arteriole and this is the efferent arteriole that is coming out. And now here it is forming a network and it is called as glomerulus. And here this one, this is the efferent arteriole. So now observe carefully, this efferent arteriole, actually it is making a peritubular network. For your understanding purpose, I am drawing like this. Observe carefully. It is actually like this. Okay, so this is the vasa recta. This is VR. VR means vasa recta. Clear? This is what the basic diagram is. Now we will observe very carefully, so that how exactly these concentrations are maintained. Concentrations are maintained. Observe carefully. Now here what they have done, up to this they made it as 300 milliosmoles per liter. 300 milliosmoles per liter. Okay, osmoles per liter. And then they made it as 600 and then 900, and at this particular one, at this level it is 1200 milliosmoles per liter. This is what they made. So now what you need to observe is, from this way, in this way you need to increase the concentration, and in this way you need to decrease the concentration, in this way you need to increase the concentration, right? Next one, for this one, in this way you need to increase the concentration, and in this way we need to decrease the concentration, because here like this concentration is increasing, like this concentration is decreasing. So observe very carefully. Okay, don't get distracted, observe very carefully. Now we will go with, here glomerular filtration is taking place, and during glomerular filtration, water, minerals, nutrients and nitrogenous waste materials are present here, and this initial one is called as a filtrate. Now filtrate is passing through proximal convoluted tubule, and in the proximal convoluted tubule 70 to 80% of these water, minerals, nutrients and all these will be absorbed, and based on the requirement some ions will be secreted. Clear? Now the remaining filtrate is coming here, but here what is the concentration? It is approximately 300 milliosmoles per liter. Clear? Next one. Now what happens? Once it is coming into this, concentration should increase. So now observe carefully. Now filtrate is having, so even though 70 to 80% is absorbed, still the filtrate is having water, some minerals and nitrogenous waste materials. Observe these things. Still it is having, don't think that 100% water is absorbed. If 100% water is absorbed, how they will flow? So some amount of water is there, some minerals are also there along with nitrogenous waste materials. So now our focus is, when this filtrate is passing through descending limb, when this filtrate is passing through descending limb like this, from here to here concentration should increase. For what? For the filtrate present inside this, because already medullary interstitium is having this concentration. So here concentration should increase. Just now we studied how to increase concentration. Increase the concentration of solution is having two options. It is having two options. What are those? By adding solute you can increase, by removing solvent you can increase. But the thing is, now I will try to use both. But only one is possible. First thing is add the solute. If you are adding solute means some nutrients, minerals you need to add. See, remember, already it is having some minerals and nutrients and water, and we need to take them. Suppose you are adding, for the increasing concentration if you are adding, and they may be lost here. So that's why here it is used. This is used. Please understand here. Here it is removal of the solvent. From where it is removed? From the filtrate. From the filtrate it is removed. From the filtrate it is removed means medullary interstitium is absorbing this. Here also some cells are there, no? And these interstitial cells are absorbing water from filtrate. That means from the filtrate water is removed. Removed means where it is going? It is reabsorbed by the medullary interstitium. So now this is a continuous process. So slowly water is keep on absorbing. So what happens? Concentration is keep on increasing. So 600, 900, 1200. Understood or not? In this way, in the descending limb of Henle loop, concentration is increased mainly by absorbing the water. Understand this. Now you can link up this information with this. Is it clear? I hope now you understood this. So now what is the concentration inside this? Here it is 1200 milliosmoles. Now here it is 600. Now here it is 900 and here it is 1200. So in this way, there is an advantage here. Concentration is increased and at the same time, water, more and more water is absorbed by the body. It is going into interstitium of kidney means what? It is absorbed by the body. Understood or not? And ultimately it will go into the blood circulation. It will go into the blood circulation. Remember this. Okay. Now in this way, from this filtrate, water is reabsorbed. Nice. Now come to the ascending limb. In the ascending limb, the direction is like this. Okay. And here what happens? In the ascending limb, what should happen? Already it is 1200. So now it should come to 900, 600, 300, right? That means in the ascending limb of Henle loop, what should happen? Concentration should decrease. Concentration should decrease. What are the options for decreasing concentration? First one is remove the solute from this or add the water. Now the thing is, observe carefully. First one, remove the remove the solute. So remove the solute means what are removed here? Na plus C is absorbed. Na plus Cl minus NaCl is absorbed. So remove the solute. Once you are removing the solute, what happens? Concentration will keep on decreasing. What is the second option? Add the solvent. At this location, suppose you are adding the solvent to this, means water to this, that water may be lost. So body is not spending this. So that's why at what location, what type of mechanism is used? Observe carefully. In the descending limb, for increasing concentration, loss of solvent means reabsorption of water. That mechanism was used. In the ascending limb, to decrease the concentration, addition of water was not done. What was done? Reabsorption of solute was done. That means from the filtrate, this solute, these are solutes, right? Na plus Cl minus act as solutes. Now these were reabsorbed by the body. That means again they are coming back into body. In this way, concentration is decreased, and once again it will get its, here it is 900 and here it is 600 and here it will be 300. Understood or not? This is what happening. And in the thin segment of ascending limb, now these are absorbed passively without spending energy. But as their concentrate, NaCl is keep on decreasing, by spending energy in the thick segment, they are absorbed by the body. So in this way, overall what happens? Concentration of the filtrate is keep on decreasing. And then now it is going into the distal convoluted tubule, and here also some absorptions are taking place, so that once again it is coming back to this 300. Here also solutes will be depending on the body conditions, right? Now the absorption, secretions, all these things are taking place. Clear? And then now it will again come back into this, and here also again concentration should be increased. How concentration will be increased? Once again here water will be absorbed. By absorbing water from the filtrate, that means inside the filtrate, water will be again decreased. So in this way, the urine formed here will be 4 times concentrated by the time it is reaching this. So in this way, water will be reabsorbed by the body. So now you can understand that how water is important. So water is important. So that's why it should be absorbed wherever it is possible. Clear? Please understand this. In this way, concentration increased. Not only this, a small amount of urea is absorbed. And why this urea is absorbed? Actually, urea is a nitrogenous waste. It should be excreted. So majority of the urea is excreted, but a small amount of urea is reabsorbed because it acts as a solute. So that for maintenance of this much of high concentration, some solute is required for interstitial cells, medullary interstitial cells. So that's why these cells are storing some small amount of urea, and this urea is used to maintain this concentration. That's it. So NaCl and urea. These are the two things that are helping in maintenance of these higher concentrations. So if more and more urea is absorbed, now excess urea will be again released into this ascending limb, so that now this excess urea can be again released. So in this way, whatever is the quantity that is required, only that much will be taken. Now excess will be again released by the body. So this is what exactly happening. So in this way, the concentrations are maintained in the nephron and also in the collecting duct. I hope now you understood this. Okay. So this is what with respect to the nephron and the collecting duct. Now we will talk with respect to the vasa recta. For the vasa recta, this is the descending limb. This is the descending limb of vasa recta. This is upside, right? So this is the ascending limb of vasa recta. Now we will observe this content. Observe the content carefully. Okay. Now here what happened? From the nephron and the collecting duct, now these water NaCl, they were absorbed into medullary interstitium first. They are absorbed into medullary interstitium first. And then from that medullary interstitium, now they will enter into this blood vessel. Now what is this one? Now this is the efferent arteriole. Actually the blood volume is decreased because some filtrate is going here, so blood volume is decreased and it should get back its own original blood volume. Now how it will do this? But at the same time, here also it should maintain this 300, 600, 900, 1200, 900, 600, 300. It has to maintain this. It has to maintain this. Now how this vasa recta is maintaining, observe very carefully. Now here, for the descending limb of vasa recta, concentration should increase. For increasing concentration, what should happen? Please remember here. Here also, for descending limb of vasa recta, concentration should increase, but it has adopted a mechanism called absorption of water. But here, for the descending limb of vasa recta, concentration should increase, but it has adopted a different one. What is that? The NaCl that was absorbed from this will enter into this. NaCl is what? Solute. Now solute is added here. By adding solute, what happens? Concentration will be increased. Simple logical mechanism. Simple logical mechanism. So the NaCl absorbed from this will be going into this. So please remember, for the normal maintenance of this concentration, whatever the NaCl required, that will be present here. That excess NaCl is entering into this. In this way, the NaCl present within the filtrate of the nephron is ultimately entering into the blood vessel. That means whatever the NaCl present here, again it is coming back here. Understood or not? So in this way, by the addition of this solute, the concentration will be again maintained. Clear? Nice. And in this way, concentration is keep on increasing. Now again concentration should decrease. How it will be decreased? A small amount of water will be entering into this. Small amount of water, the water that is absorbed here, that is entering here. So here what happens? By the addition of water, it will be diluted. So concentration will be decreased. So in this way, the water and the minerals, the nutrients, whatever they are lost here, all these are again coming back. They are again coming back into the blood vessel. That means technically what are removed here? Nitrogenous waste plus very small amount of water is released out, and remaining all the things, even though they got filtered here, ultimately they are coming back here. And in the NCERT they even said that a small amount of NaCl is going out. Even loss of solute also will decrease the concentration, right? Small amount of NaCl is again going back into interstitium here. Though it is entering here, some small amount is going here. This is again to maintain the concentration of medullary interstitium. So now here, how much NaCl will go and whether it will go or not, and all these things are based on the conditions here. If this is maintained, now it don't need to go. So now it is going. If it is keep on flowing like this, after some time this concentration is slightly decreasing, then NaCl will come out here and water is entering here. So ultimately they will try to maintain these concentrations. Anything excess than required, now it is entering here and it is going along with the blood. So in this way, what is exactly happening? Nitrogenous waste materials are removed along with small amount of water, and the remaining major quantity of water, minerals, nutrients, all of them from this, first they are entering into medullary or cortical interstitium, and then again they are coming back into the blood vessel only. So in this way, what is the ultimate conclusion? The blood that is coming to the nephrons is ultimately removing the nitrogenous waste and getting back everything. Only nitrogenous wastes are removed. So in this way, kidneys means nephrons are involving in filtration of the blood, so that they are removing the harmful nitrogenous waste materials. This is what the ultimate conclusion is. If you don't understand this interlinking, then there is no use in by hearting this particular one. Clear? And this movement of this particular fluid in these different tubes in opposite directions is called as counter current mechanism. Clear? And this much was mentioned in NCERT. I hope you understood this particular concept, and the same thing in NCERT they have represented here. So from where it is going, there where it is entering, observe this carefully, you will understand this. Clear? Not only this, some extra locations also they have mentioned here. Water is removed, vasa recta, not only NaCl entry, even small amount of water is absorbed back, so that concentration will increase. And here, so NaCl small amount is absorbed back. Not only this, water is also entering into this, so in this way it will be diluted. So you can understand this. Simple. Ultimately you need to understand that how exactly concentrations are maintained. Focus on this. Where solute is added, where solute is removed. Where water is added, where water is removed. Understand on this particular point, that's it. Entire mechanism you can understand in just 2 minutes. Just 2 minutes. Clear? This is what. In this way, we are talking about this filtration, concentration of the filtrate. So ultimately, so when talking compared with 300, at the end of this collecting tubule, 1200 is there. So this will be 4 times concentrated. That is what the meaning that they are saying. Clear? Next one. Regulation of the kidney function. Here they are talking about total 3 mechanisms. First thing is when blood volume decreases or blood pressure decreases or glomerular filtration rate decreases, then what happens? There are 2 mechanisms. Now antidiuretic hormone, also called as vasopressin, actually it is produced by hypothalamus and released from neurohypophysis. Yes or no? Posterior pituitary. So it will be released from neurohypophysis. So now it is acting on the lateral parts of the tubule, means where distal convoluted tubule and collecting duct, there it is acting. And since our body requires more water and Na plus, now they are involving in absorption of more Na plus and water. Now this absorbed one again ultimately going into the blood vessel only, so that again the blood volume will be maintained. That's it. Simple logic, that's it. In this way blood volume will be maintained. When enough blood volume is maintained, now this release will be stopped. That's it. In this way ultimately it is in helping in maintenance of blood volume, maintenance of blood pressure, maintenance of glomerular filtration rate. Next one is renin. Renin from juxtaglomerular apparatus. Just now we have studied about this. Now this renin will convert angiotensinogen into angiotensin 1, and that will be converted into angiotensin 2, which is acting as a vasoconstrictor, so that blood pressure will be improved. Because of the less blood volume, the blood pressure got decreased. Now because of vasoconstriction again, blood pressure will be increased, so that glomerular filtration rate will be increased. So ultimately it will cause the retaining of again normal blood volumes and blood pressures and normal GFR. That's it. And here not only this, and this angiotensinogen 2 not only a just vasoconstrictor, it is also acting on adrenal cortex, so that it will cause the release of aldosterone, that is a mineralocorticoid. So it will help in the again reabsorption of Na plus, that will also ultimately cause increase in the blood volume. So this is how it will be useful, and it is simply called as renin angiotensin mechanism. So in this way the blood volume will be retained. Suppose this blood volume is keep on increasing. When blood volume is keep on increasing, now this blood is ultimately going to the heart only. In the heart, the atria, they are feeling this more blood volume. And now from this atrial wall, now there is a factor that is released. It is called as atrial natriuretic factor. What is this natriuretic? Natrium means sodium. Na is sodium. What is that Na full form? Natrium. So that natri and uretic, excess water will be released in the form of urine. So Na plus and water, they will be released in the urine. So that's why it is called as natriuretic factor. Now it will cause the loss of or the release of excess Na plus and water from the body. So that's why it will try to decrease that volume so that it will be maintained. So when blood volume is decreased, these will act. When blood volume is increased more than required, now this will act. So in this way, all these are ultimately helping in maintenance of blood volume, blood pressure and glomerular filtration rate. Very clearly they mentioned in NCRT, you can simply go through this. Next one is regarding the micturition. The release of urine is simply called as, if urine from the body is called as micturition. And here what happens, now from that kidneys, the urine will be collected through the ureters into the urinary bladder. Now these are the openings of these two ureters, and now this is where the opening, and through this opening now this will be released out. But the thing is, initially the urine will be collected within this urinary bladder, so that now it will be keep on stretching here, and here there are some receptors called as a stretch receptors are present in this muscular layer. And when urine is filled in enough quantity, now they will cause or they will exert some pressure on these walls, so that the stretch receptors will be activated. They will send the information to the nervous system. So from the nervous system the information is coming back, so that it will cause constriction. That means it will cause the contraction of this particular urinary bladder, and at the same time it will cause the relaxation of these sphincter muscles, so that the urine will be released out through urethra. This is what they are talking here. Clear? And this neural signal related to this particular process is called as micturition reflex. That's it. It's very simple information they have given in NCRT. And here they are given some data, so that's why I have directly kept this particular one. Now let us see. The process of release of urine is called as micturition, and the neural mechanism causing this is called as micturition reflex. And then an adult human excretes on an average 1 to 1.5 liters of urine per day. Even though 180 liters per day filtrate is formed, but still just 1 to 1.5 liters of urine is produced. That means approximately 99% is reabsorbed back. Next one, the urine formed is light yellow colored, and it is a watery fluid which is slightly acidic because it is having pH of approximately 6, and it has its own characteristic odor. And on an average 25 to 30 grams of urea is excreted out per day. Each day this much of urea is excreted. Not, please remember, even though human is considered as ureotelic, we are excreting uric acid and ammonia also. Along with this, many other excretory materials we are releasing. So please remember this. Various conditions can affect the characteristics of the urine. So in different disease conditions, in different conditions, and what type of food that we are eating, so based on this the characteristics of urine can be changed. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. Suppose an individual is already having metabolic disorder. So these metabolic disorders are due to some genes and all, right? So we studied about that phenylketonuria in principles of inheritance and variation. Phenylketone compounds are excreted along with the urine, right? So those things also you can understand based on this analysis. Not only this, malfunctioning of the kidney. Suppose kidney is not functioning normally, then also you will find some different components in that urine. Based on this you can say that kidney health is not good. This is what they are saying. For example, presence of glucose in urine is called glucoseuria, and presence of ketone bodies in urine is called as ketonuria. And if we are observing this, now these are the indications of diabetes mellitus. Yes or no, the sugar disease that we are saying, no? So they are the indicative of this diabetes mellitus. So in this way they can give a particular disease condition also. Next one, role of other organs. Not only kidneys, there are some other organs like liver, skin and lungs. They are also involving in release of some of these components. They have given some information. Let us quickly read this. Our lungs remove large amounts of CO2, that is approximately 200 ml per minute. Per minute they are asking, so they can ask for per hour, per day also they can ask on an average value. So you need to able to multiply all these things. Okay, so large amounts of CO2 and also significant quantities of water every day. Please remember, when we are breathing, doing this breathing, inspiration and expiration, that air is humidified. That sentence we studied in that breathing and exchange of gases. Humidified means some amount of water is going into that. So when we are releasing the air, so some amount of water will be lost. So we are losing some amount of water in that form also. That is the reason why, suppose a person is keep on talking continuously, so we are getting the dryness of this mouth and the throat and all, because some amount of water will be lost when we are speaking like this along with this air. So please remember this. Next one. Liver, this is the largest gland in our body. This is secreting some components like bile containing substances like bilirubin, biliverdin, cholesterol, and degraded steroid hormones, vitamins, and even drugs also. Now it is liver is excreting these many, but liver is releasing all these things along with the bile, so that now these things will be released out along with the undigested food material, along with the fecal matter it will be released out, okay, not through the kidney, okay. Next one. The sweat and sebaceous glands in the skin can eliminate certain substances through their secretions. Sweat glands, they are releasing the sweat. Sweat means it is mainly having the water, and the sweat is a watery fluid containing NaCl. It is having some minerals also, and small amounts of urea is also present in that sweat, and lactic acid is also present. So in this way many components are present, but please remember, sweat glands main function is not the excretion. The primary function of the sweat is to facilitate the cooling effect. Suppose you are going into the sun, suppose the temperature is keep on increasing, then body temperature, the surface temperature should be decreased. At that time this sweat will be released, and this sweat is having water. So when this water is getting evaporated, it will give the cooling effect. That is the primary function. But along with that, it is releasing some other components, and even it is having some nitrogenous waste material also in it. This is the point that they are saying. And next, sebaceous glands. They are eliminating certain substances like sterols, hydrocarbons, waxes through the material that is called as the sebum. Sebaceous glands will release sebum, and sweat glands will release the sweat. These two are completely different. And this sebum and this secretion provides a protective oily covering for the skin. Okay, so it will prevent the dryness of the skin. This is what they are saying. And here they are asking a question: do you know small amounts of nitrogenous waste could be eliminated through saliva also? Even in the saliva also, small amount of urea is removed. So please understand this. Next one, they are talking about the disorders of the excretory system. And first one they are talking is regarding the uremia. What is this uremia? Increased concentration of urea within the blood. Normally in the blood, some allowed concentration of urea is present. If that concentration is increased more than required, then it is called as uremia. Why it is increased? Because kidneys are not filtering properly. There is a problem with filtration at the kidneys. There may be some infection, or they may got damaged, or something might have happened. So that's why this urea concentration is increased. When urea concentration is increased in the blood, now you need to remove this. Not only this, there are some other components like creatinine and many things will also increase. Now we need to remove these nitrogenous waste materials. So we need to do the dialysis of the blood hemodialysis. Blood should be undergoing dialysis for the removal of these nitrogenous waste materials, and this process is called as hemodialysis, and this is done with the help of artificial kidney. In the case of artificial kidney, what they will do? So they will take the blood from the artery. Please remember these things. These words are very important. From the artery we are collecting this blood, and this collected blood is connected to this artificial kidney. In the artificial kidney, what happens is the blood is passed through a tube like coiled tube like structure like this, okay, and this is how it is having the opening, suppose. And now surrounding this there is a chamber, and this chamber is filled with a isotonic solution, but it is not having, there is no nitrogenous waste. It is filled with a fluid, and this fluid is having exactly same concentration as that of the blood, but this fluid is not having nitrogenous waste materials. And now the blood from the convenient artery, now it is passing through this. Blood is passing through this. For all other components, it is having exactly same concentration. So for those things, there is no movement. But blood is having nitrogenous waste, but this fluid is not having nitrogenous waste. So from high concentration to low concentration, they will come. That means from the blood, nitrogenous waste will move into this fluid. So intermittently this fluid will be removed and the fresh fluid will be used here. So in this way, the nitrogenous waste materials will be removed from the blood. And finally this blood will be again given to this artery. To the, so not to the artery, into the vein we need to connect this. Blood is collected from the artery. From the artery we are collecting this, and ultimately it should be given to the vein. So this is what the mechanism that we are doing. And in this process, you need to remember another point. When you are taking the blood out of the artery, there is a huge chance that it may undergo coagulation or clotting. If blood is undergoing clotting, then what is the use of this? So blood should not clot. So that's why here we are adding heparin, which is an anticoagulant. Even though heparin is already present within the blood, that is not enough to stop this coagulation. So we are adding some excess heparin to this. So when we are before sending to this, as soon as it is coming out of the artery, so we are adding heparin so that the blood will be in fluid condition only. It will not coagulate. And then it is after this diffusion process and all, and finally it should be released into the vein. And before releasing into the vein, we need to add anti-heparin. So whatever the heparin we have added here, so that should be nullified here, and then the blood will be again sent back into the patient through the vein. And this is how we are ultimately removing nitrogenous waste materials, and this is called as artificial kidney. This is what they are saying. Okay, and this fluid is called as dialysis fluid or dialyzing fluid, anything you can say. This is what they are saying. And if for a normal, suppose kidney is having some small problems, and if you want to give some rest for few days, so that during that time you can go for this artificial kidney, so that the load on the kidney will be decreased, so you are giving some scope for healing of that kidney. Suppose the kidney is completely damaged, then that is not useful, and every time we cannot go for the dialysis because it is costly. So there is another alternative, that is the transplantation. So before going for the transplantation, we need to go for antigen matching also. That's the reason why generally they say that, so matching will be more better or more successful if the kidney is taken from close relatives. So that's why from the close relatives we can take a kidney, and that kidney can be transplanted here, so that the chances of rejection will be very, very less. This is what they are talking here. This is what the ultimate concept of this one. And finally they are talking about renal caliculae. Sometimes what happens is some oxalates along with the calcium and some other components, they will accumulate, and finally they will become strong structures like stones, and these are called as kidney stones or renal caliculae. Okay, and not only just in the kidney, so they can be formed in different other locations also. So there are more problems with these renal caliculae and all. And the thing is, generally those individuals who are not taking enough amount of water, so in those individuals there are more chances of formation of this renal caliculae. Next one is glomerulonephritis. So it is the inflammation of glomeruli of nephron or kidney. Okay, this is what they are talking. So with this, the NCERT content for this particular chapter is completed. And before concluding this, once again I'm telling you, so these revision videos are available on YouTube channel and also on Live Prep app, but test series can be given only through the app. So with this part is completed. In the next lecture we will meet with a different chapter. Thank you.